Hi, my name is Filipe Figueiredo, and until recently, I was a PhD student at both the Charité Universitätsmedizin Berlin and the Instituto de Medicina Molecular Jolo Bentons. For the past few years, I've been doing my PhD project on mitochondrial DNA mutations in patients with multiple sclerosis and trying to see if there was a link to disease activity and progression. This was possible due to the financial support from our four funders, the Fundação para a Ciência e Tecnologia, the Programa Operacional Regional de Lisboa Portugal 2020, the National Multiple Sclerosis Society of the United States of America, and Merck Germany. Multiple sclerosis, or MS, is a chronic autoimmune disease that affects the central nervous system, that is, the brain and spinal cord, causing demyelination, which is a damage to the myelin sheaths that envelop the axons of the neurons. Worldwide, there are around 3 million people living with MS, with a new diagnosis being made every 5 minutes. The exact cause behind the disease is unknown, but it appears to be autoimmune in nature, where we observe an increase in pro-inflammatory cells and a decrease in anti-inflammatory cells. Although other cell types are also relevant, CD4 T cells are thought to be crucial for this process. The cause behind this autoimmune dysfunction is unknown, although it appears to be multifactorial, with a contribution from both the environment, which includes Epstein-Barr virus infection, low vitamin D levels, obesity and smoking, and genetics, which includes both nuclear and mitochondrial DNA mutations. In this project, we focused on mitochondrial DNA. Our research question was then built based on 3 key pieces of information. Brain samples from patients with MS show mitochondrial dysfunction, mitochondrial DNA mutations have been associated with MS risk, and CD4 T cells from patients with MS have impaired mitochondrial activity, which might lead to an increase in pro-inflammatory cells. What we didn't know was if this mitochondrial dysfunction was caused by the mitochondrial genotype of these cells. To answer this question, we developed a project focused on the mitochondrial genotype of CD4 T cells in patients with MS, trying to link it to disease activity through time. We had 2 approaches: a cross-sectional approach, where we looked at newly diagnosed patients and age and sex match healthy controls, and a longitudinal approach, where we looked at the same patients 30 months after their first clinical visit. Since we were working with very small amounts of cells and thus DNA, we used a next-generation sequencing library mostly aimed at forensic research and suitable for very low amounts of DNA. However, this method was only able to identify mutations that were prevalent at 10%, which is somewhat high for next-generation sequencing, and with a different reference sequence. For this reason, we developed a bioinformatic method that was able to circumvent these issues and that we recently published. With this method, we were able to identify more mutations at a lower threshold with better performance, even though we were losing some information due to the quality control method we employed. In our MS cohort, we were then able to use this method to get adequate and uniform results from our samples. We first started with a cross-sectional analysis, where we had 3 different approaches. The first approach was looking at all mutations that we found in comparison with the reference sequence we had. The second approach was looking at mutations that were predicted to affect the function and/or structure of proteins, so-called deleterious mutations, whilst also looking at the cumulative burden these mutations had. The third approach was similar to the previous one, where we look at mutations predicted to affect the structure and/or function of tRNA, which are important for protein assembly. Overall, we did not find any statistically significant differences between the different subject types. We did see, however, a reversal of the mutational patterns between patients and healthy controls. While healthy controls were overrepresented for all mutations, patients with MS were overrepresented for deleterious mutations. We then looked at longitudinal mutations in patients with MS, particularly at mutations that were only present in one of the clinical visits, and at changes in prevalence for mutations present in both visits. Overall, patients with MS had stable genomes longitudinally. Finally, we explored the link to disease activity and progression, where, as a positive control, we first confirmed that the existing variables for clinical and imaging visibility associated with different diagnosis trajectories. However, when we did the same analysis for next-generation sequencing variables, we did not find any association. Thus, although we were able to successfully sequence the mitochondrial genome from our samples with a novel method that we developed, it does not seem that the previously reported mitochondrial dysfunction in CD4 T cells is caused by their mitochondrial genotype. Further research should look at transcriptomic and proteomic changes, ideally with single-cell sequencing and beyond the CD4 T cell compartments.